So these are notes on Introduction to Protein Networks. So we've been talking about how enzymes change things in our body. So in our last lab, we looked at how lactase, the lactase enzyme, changes lactose into galactose and glucose, and that not having that enzyme means your body isn't able to change the lactose into galactose and glucose. You can't break it down the way you need to, and we would call that being lactose intolerant. And one of the things we've talked about is the fact that enzymes are proteins. So we have these networks of proteins in our body that change one compound into another compound. And those compounds, the substrates, remember, could be carbohydrates, they could be other proteins, they could be lipids, they could be all sorts of things. So the first thing we're going to look at are pigment molecules. So a pigment is a compound in an organism that gives it some characteristic color. So like chlorophyll is a pigment that gives plants a green color. Here we're going to have an imaginary plant called a round bud. It's not a real plant, but it'll give you an idea. And so round buds have this characteristic red color here, and that's caused by some pigment. Melanin that gives our hair and our skin certain colors, that's a pigment. And often pigment molecules are catalyzed to change their configuration by enzymes. So they're often substrates for enzymes. So we're going to look at this imaginary flower called a round bud. And round buds, we're going to imagine in our experience, have always been red. Every round bud we've ever seen has been red. Until some really knowledgeable, observant teenager, possibly you, discovered a meadow full of white flowered round buds. Okay, so up until this point, let's imagine every round bud flower we had seen had been red, but now we have these white round buds. So now we have some variation in this trait. And we're going to look at how protein networks help us to understand where this variation comes from. So let's suppose here, and let me get my pen just because I like to have it ready, that we now understand that there are white round buds and red round buds. And as we do a little more digging and kind of look at chemical makeup of these flowers, and you can do this with spectroscopy or chromatography, lots of different ways to figure out what chemicals are in different substances. We find that there is a colorless molecule which makes flowers white, and that there is a red molecule which makes flowers red, and that the white molecule becomes the red molecule with the help of enzyme Q. Okay, we're not going to worry about, this is a kind of a made-up example, so we're not going to name enzyme Q. Later I'll show you an actual example, and we'll put names on these enzymes. But remember that each enzyme is specific to the reaction it catalyzes. So if enzyme Q is changing white to red, it's not also changing, say, lactose to galactose and glucose. That would be the lactase enzyme. So each enzyme is unique to the reaction it catalyzes. So we have this colorless molecule here, and that fits into the active site on enzyme Q, changes to a red pigment molecule, and now the flower is showing this red color. So let's look at what happens if enzyme Q is not present. This isn't huge rocket science. Hopefully you see that without enzyme Q, no enzyme Q, the colorless molecule stays colorless, and you would have your white flower. So we can extrapolate or conclude or kind of put 2 and 2 together and hopefully come up with 4 and say flowers that for whatever reason are producing enzyme Q end up red. Flowers that for whatever reason don't produce enzyme Q end up white. And later we're going to link this back to genetics. Some of you, I'm sure, are already kind of putting 2 and 2 together, maybe and coming up with 3.5 in your head and saying, oh, wait a minute, these could be different genotypes if I make enzyme Q or not make enzyme Q. If you're not quite there yet, don't worry, we're going to get there. We're going to talk a little bit more about protein networks first, though. So let's look at another imaginary flower. We're just going to call this some imaginary bioflower. And here we have blue flowers and purple flowers. So it made sense before when we had this colorless pigment that the enzyme changed to a pigment that had color. But let's look at now a longer protein network than just one thing. We talked about lactose, which there's just one enzyme, lactase, that converts it. But sometimes there's more than one enzyme. So let's look at this. We're going to, again, have a colorless starting molecule that's converted by enzyme X to a blue pigment, and then enzyme Y converts the blue pigment to a purple pigment. So we could write a pathway like this. Colorless compound, enzyme X makes a blue pigment, enzyme Y makes a purple pigment. If we, for example, are producing enzyme X but not enzyme Y, then we would end up with a blue flower. If for whatever reason we also weren't producing enzyme X, then we could end up with a third choice, a colorless flower. And so you can see how having or not having different enzymes can actually change the physical characteristics of an individual. Why is it thinking? Okay, so here we go again with the pathway. So colorless white molecules converted by enzyme X to a blue pigment molecule, converted by enzyme Y to a purple pigment molecule. Here we'll have a little cartoon. You can see that these are two different enzymes. They have different active sites. They have different shapes. So let's see what happens. So there we go, converted to a blue molecule. That is now converted to a purple molecule. We have a purple pigment. And if anybody's interested, I can talk about like how different molecules absorb and reflect light differently. If you go back to chlorophyll, remember that the chlorophyll molecule absorbs all colors of light except green and yellow, and it reflects the green and yellow back, and that's why we see it. But if you change that molecule around, it's going to have different properties, and it's going to absorb different wavelengths of light. So that's how we're getting these different color molecules. It's not that the molecule changes color, it's that it changes how the light interacts with that molecule. If we don't, for example, have enzyme Y, we simply end up with a blue flower. I thought that was supposed to come out of there. Oh well. Oh, there it goes. All right. All right, so now let's look at a real life example. Let's look at serotonin. Serotonin is a molecule in our bodies. It's a neurotransmitter, meaning it is transmitted between neurons in our brain. The neurons are, of course, the cells in our brain that help us communicate, but they communicate by these things called neurotransmitters, by these chemicals. So having or not having certain chemicals helps regulate all sorts of things. And serotonin, one of the things it does is it regulates emotion. And if you have very low amounts of serotonin, that can actually cause depression. So I actually knew somebody, a biologist friend, who's going through a really hard time, and another one of our friends bought her a little necklace that was the chemical structure of serotonin. Kind of be like, hey, don't be depressed. Here's some serotonin for you. Little biology dork humor, but also a very nice gesture. So let's look at how serotonin is produced, and then how we can use our understanding of how it's produced to help treat depression, other than buying people necklaces that have the chemical structure of serotonin on them. All right, so remember that your proteins being the enzymes that catalyze these reactions, these changes, are what determine your characteristics. And on this page, there are 3 different enzymes: TPH, AAD, and MAO. Please do not freak out. I am never going to quiz you on names of specific enzymes. So I care more about that you understand what a protein pathway is, and how it can affect characteristics, then that you remember that tryptophan, for example, which by the way is the compound in turkey that can make you really tired, that tryptophan is converted by TPH into 5-HTP. I don't really care that you memorize that, although I would write down the pathway so that you have it and you can understand. So we have tryptophan is converted by TPH into 5-HTP. That's converted by AAAD into serotonin. Serotonin then is converted by MAO into 5-HIAA. And you could certainly look up what each of these compounds does, but it's the serotonin here that helps to regulate the emotions, and so we're going to talk about that. So let's look at if you had low amounts of AAD, would you have more or less serotonin? So you may want to pause the video and actually think about this before you see the answer. So go ahead and do that. Come up with your answer, more or less. So if we don't have AAD, we would expect we would have less serotonin because the AAD is converting the 5-HTP into serotonin. So less of that enzyme, less serotonin. If you had low amounts of MAO, would you expect more or less serotonin? So not as much MAO, you're going to have more serotonin because the serotonin is not being converted into 5-HIAA. What if you had high amounts of MAO? Would you have more or less serotonin? So if you had more MAO, you would expect less serotonin. So the question is, do you want lots of MAO or a little bit of MAO? And think about your answer. Again, pause the video, think about this. Think about why, not just an answer, lots or a little, but why. So realistically, you want, I would put this in quotes, just the right amount of MAO. If you have too much MAO, you're going to be susceptible to depression. You're not going to have a lot of serotonin. But on the flip side, if you don't have enough MAO and you actually have too much serotonin, that can actually lead to almost the opposite of depression, like an extreme giddiness. It can lead to racing heartbeat. It can lead to so a bunch of symptoms that you also want to avoid. So you want the right amount. But people in general, it's much more likely to have too much MAO than not enough. So it's much more common to be depressed than to have, it's called like serotonin syndrome or something. Most of us have never heard of this because it's just really not that common. But what is a lot more common is depression. And that is caused by having too much MAO. And so we have these drugs that we've made that are called MAO inhibitors, or you've maybe heard of MAOIs. If you start listening to like drug commercials on TV when they're advertising a new drug and they're saying, ask your doctor about this, a lot of times they'll say should not be taken by people taking MAOIs. So an MAOI is an MAO inhibitor. It's going to block the MAO active site, and so the serotonin can't bind to the MAO. It doesn't take away the MAO, but it blocks that active site, so the serotonin can't get in and be converted to 5-HIAA. And so then people with depression can take MAOIs, too many letters, I'm getting all jumbled up in my head, to increase their levels of serotonin to combat their depression. So this is how understanding the entire protein network can help us treat a disease like depression by targeting the enzyme that turns serotonin into something else in our bodies. So please feel free to add questions to the comment section of YouTube or ask them in class, and we're going to work a little more with protein networks in class so that you really start to get this. Have a good night.